All right, good morning, everybody. Today is Wednesday, September 16th, 2015, and this is Before the First Cup with Jules here on UCY.TV. Happy Wednesday, everybody. We've made it halfway again. It is 69 degrees and partly cloudy here in upstate New York. It's supposed to be a warm one again today, so that should be nice instead of fall. I mean, I like fall, but hey, I'm willing to get another week out of summer here before the calendar actually changes. Well, before we get to where we left off on Monday, I want to give a couple big shout-outs to a few donations that we've received over the last week. I want to give a huge thank you to Uff in chat. Thank you so very much, Uff, because it definitely will help. I have some hard drives I have to replace here for our archives. I also want to give a huge shout out to Ms. Lonnie Clark. Thank you, Lonnie, because that was extremely generous of you. And again, hard drive. I need a nice big fat hard drive. And also we received a donation from Dambo. And I'm not quite sure who Dambo is, but I know Dambo must be a listener, and I want to thank you extremely from the bottom of my heart. Thank you very much to everybody who has donated to UCY, because without you guys, we wouldn't be able to continue what we do. And UCY, honestly, I got to tell you, it's grown a lot, even though it doesn't seem like it sometimes. It has, as far as our worldwide reach. And if you just take a look at the show count plays, you can see that our audience has definitely grown. So I want to give a huge thank you to everybody, contributors and listeners and people who share our information. All right. So where do we leave off on Monday? Because we dug into some pretty disturbing information. Actually, the last couple of weeks have been extremely disturbing for people like me who have a thing about their food, and every single nightmare I've ever had appears to be coming true between clone food and mad cow disease, and we still got to get back to the mad cow. Maybe we'll hit on that tomorrow. Some really horrifying things that I found that happened in 2008 that apparently got swept under the rug. But on Monday, we left off looking at edible vaccines, genetically modified plant material that have been engineered to produce antigens. And where we first heard about this was in the quote-unquote secret serum for the Ebola outbreak. Oh, my God. But I had heard about this even before that, back in 2006, when Bush was still in office. They had launched their Project BioShield, and we were told in a training class that I took that was sponsored by FEMA that they were working on the universal flu vaccine, and that was being genetically produced by modifying plants to be able to produce instead of eggs. And it would use your body's immune system to have a signature for a flu-ish like virus, let's say, and then be able to search and destroy. So anyway, let's dig a little into where we left off on Monday. We titled the show, Edible Vaccines in GMO Bananas, Tomatoes, and Potatoes with a question mark. And yes, we read all kinds of stuff that had looked at some of the research that was done starting back in the early 90s with the banana, and maybe even in the 80s. I don't remember how far back that went. I believe that they abandoned the banana and they went into tomatoes and potatoes, and they have both been tested in the general populace. We also dug into some of the chronic wasting disease and its connections with Alzheimer's, mad cow, aka bovine spongiform encephalopathy. Yes, I do like to say that. Scrapie and some other brain wasting diseases that were very similar in the fact that they all had something in common with malformed prions. And then some new news came out last week that they now believe that it is potentially possible, maybe not person to person, but definitely through bodily fluids and surgeries, let's say, for those malformed proteins that they found in Alzheimer's, those are the amyloids, to be able to be passed from person to person with internal fluids. So they were saying blood transfusions, brain surgeries, and then the story that came out Friday, I think it was Friday, that we talked about on Monday, was through dental surgery as well. So ding ding, be very careful. They always had issues with trying to properly decontaminate surgical instruments of prions, because prions, like I said, are not alive, they're proteins. And so standard methods of sterilization did not seem to be working as well as they thought. And like I said, sounds very similar to the DOD and some of their issues with trying to kill anthrax with radiation that they found out many years later hasn't been working, and they've been shipping it all over the planet. I want to continue just a little bit more on these edible vaccines, because there were a few other things that we ran across besides the GMO banana vaccine, tomato vaccine, a potato vaccine, and one of those was the rice vaccine. This is out of June 2nd, 2014 in the New York Times. Yoshiyasu Yuki and other researchers at the University of Tokyo are bioengineering rice in a bid to turn it into an easy, low-cost storage and delivery medium for drugs to combat common infectious and contagious illnesses. The immediate target is to develop new treatments against cholera and rotavirus, two causes of severe and often fatal diarrhea. And we actually heard that in some of the stuff that we were reading on Monday as well, is diarrhea was the big thing that they were going to genetically modify plants for so they could grow them in third world nations and give the vaccination to the poor children without them pretty much knowing. Cholera now kills as many as 120,000 people a year, according to the World Health Organization, while rotavirus is estimated by the organization to kill about 500,000 children a year under the age of 5, amounting to about 5% of all child death worldwide. And hey, you guys know me, I'm the humanitarian. I am not against protecting children from dying from things, but what I am against is mad science gone mad, and some of the stuff that they're doing right now and getting into people who don't know any better to then find out 20 years later that, whoops, it causes an arm to grow out the back of your head. You know what I'm saying? Like, this is definitely leading edge science that if we had benevolent people at the top of the food chain, I'd be all for this, but we don't. And so for every right side of the coin, there's the back side of the coin where things can be used for evil, things can be slipped into our food supply without our knowledge, especially when we find out that the USDA and the FDA aren't keeping us safe, and that our Congress has voted to get rid of labeling laws. You know, there seems to be an agenda afoot here. You look at United Nations and we're running out of food, and oh my God, the carbon quotient and beef is just a big carbon sink, and it's got diseases, and we can't feed the world, so we have to find an alternative. I mean, there definitely is an agenda here. Vaccinate All Children Act, Healthy People 2020 Act, yep. Nobody wants to see a needle coming, so we're going to give it to you whether or not you want it. Vaccines or antibodies for both exist but require refrigerated storage, and that seems to be the big echo that I keep hearing here is the reason for breeding vaccines into food, is it's all about needing refrigeration. And that also came out during the Ebola quote-unquote outbreak in Africa. Because it was hot there, they needed to deliver the vaccine quickly on ice and get it into the first responders, I believe, that they were injecting it into. Anyway, vaccines or antibodies for both exist but require refrigerated storage. Mr. Yuki, an assistant professor of mucosal immunology, said in an interview. Mucosal immunology is interesting because I've also seen and heard that echoed a few times now as well when I've been digging into this. It seems to be that one of the easy ways to get these antibodies into the body is through the digestive system, and by putting it into food, it becomes encapsulated, and it basically sneaks by your immune system into your digestive tract, where it can then be directly absorbed. Back to the story. Bioengineering vaccines or antibodies into rice would allow them to be stockpiled easily without the cost of cold storage for up to 3 years at room temperature, he said. The rice could be ingested orally, ground into a paste, and drunk, delivering the antibodies to the intestine, he said. Mr. Yuki said that his team figured out how to make a cholera vaccine using rice in 2007, and a rotavirus antibody last year. The rotavirus treatment was developed from highly stable antibodies found in South American llamas that are uniquely resistant to heat and acid in the animal's stomach, says Lennart Hammarström, a professor of clinical immunology at the Karolinska Institute in Stockholm, who co-authored a paper with Mr. Yuki's team on the research program. Mr. Yuki said the team immunized a llama against rotavirus infection, extracted and decoded its DNA, and engineered a strain of Ninbare rice, that's Nihonbare, with the same DNA blueprint. Adding a storage protein synthesis inhibitor into the genome, the team produced rice with a high yield of antibody content. So they actually used a llama, and they got the llama. Did they infect the llama with rotavirus? A team immunized a llama against rotavirus. So they gave the llama some sort of injection, I'm assuming, to immunize them against rotavirus, and it does not say what kind of injection that is or anything. Then they extracted and decoded the llama's DNA and engineered a strain of Ninbare rice with the same DNA blueprint. So they must have decoded the DNA to find where that DNA that gave them the immunity was, and then somehow slipped that immunity into the rice DNA blueprint. That's very interesting. And then added a storage protein synthesis inhibitor, which I have no idea what that is, into the genome. And the team produced rice with a high yield of antibody content. 1% of the total weight of the rice grain is a rotavirus antibody, said Mr. Yuki, and that is phenomenally high as a percentage. Both the cholera vaccine and the rotavirus antibody version of the rice have been tested on laboratory mice, Mr. Yuki said. Clinical trials of the cholera vaccine are planned to start in a small group of people next year to test for safety and possible side effects and establish dosage ranges. And this was from 2014, so somewhere in the world they are now doing clinical trials on injecting on GMO vaccine rice. It says the first phase of trials should be completed within 2 years and could be followed by several years of larger trials, such as countries as, yes, Bangladesh, where cholera is a major public health threat and where there's lots of people who won't know any better. Plant-based drugs, however, are a rarity. Sure, except for the testing of the banana, the tomato, and the potato, says Fumio Tawikawa, senior researcher at the Functional Transgenic Crop Research Unit of Japan's National Institute of Agrobiological Sciences. Wow, that was a mouthful. And to clear the regulatory hurdles to bring this medication to market may be difficult, he said. Well, it probably won't be, I'm going to interject, probably won't be in the United States with our FDA being bought and sold by Big Pharma and Big Agra. Mr. Yuki conceded that it could take at least 10 years for his vaccine to reach the market. Still, making drugs using plants can drive down the prices dramatically. And of course, yes, that is the fuel behind this, because they want to test all this newfangled medicine on the countries that can't afford it. back to the story, because much of the price of drugs can be attributed to factory processing costs that would be bypassed in a plant-based medicine, right? And our research and development. So if we don't really have to pay for research and development, and we can be doing it in third world countries, well, then it's going to get rid of a big line item budget. Now, wouldn't we know it, this next paragraph just tells the entire story. Despite the possible regulatory challenges, Mr. Yuki says that, yes, ding, 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 Bill and Melinda Gates Foundation has shown interest in the cholera vaccine, and a couple of pharmaceutical companies were showing interest in developing drugs based on his team's research. He, of course, declined to name them, citing his non-disclosure agreement. Well, didn't we find out who's jumping on the bandwagon? And yes, Bill and Melinda Gates, who already had paid for research for putting the malaria vaccine into goat milk and giving it to small African nations. Yes, that same Bill and Melinda Gates Foundation. Mr. Yuki said that his team was also considering setting up a life sciences venture called Muco Biotech, M-U-C-O Biotech, to take on the commercial development and marketing roles. He said his cholera vaccine now costs about 1,000 to 2,000 yen, or about $10 to $20 for a treatment course of 3 doses of about 3 grams each, produced at trial scale. But it could come down to about $1 to $2 with mass production. And as a cholera vaccine and adult treatment, yes, look out, would be about 10 to 20 grams of rice grains ground into a powder, dissolved in a cup of water and swallowed. He says, I'm not sure how it tastes, but the taste doesn't matter. No, they could mix it into anything. But if not to add insult to injury, there's more. Yes, not only is it bananas, tomatoes, potatoes, and rice, but now we got a 5th one in the rankings, lettuce. This one is out of December 2nd, 2013, and hold on one second because there's audio with this one. Yes, they are developing antibodies in lettuce in Pennsylvania. Lettuce, the new secret weapon for making vaccines cheaper in the developing world. Here we go. Maybe. Hold the phone. I am connected. I have audio. It's just not playing through Skype. All right. You know what? While this is trying to load, let's read a little bit of the story. It says, in a brightly lit lab in West Pennsylvania, rows and rows of tiny lettuce plants are sprouting in clear containers, stretching their thin roots into a clear growing liquid. Located within Penn State School of Dentistry, which I thought was kind of odd. Why are we doing GMO lettuce plants to give vaccines in the School of Dentistry? But whatever. Located within Penn's School of Dentistry, the lab is run by Henry Danielle, a professor in the Department of Biochemistry and Pathology and director of translational research. Danielle wears a colorful DNA helix tie, and his lab seems abuzz with lights, ventilators, and his energy, and he hopes that lettuce can be a secret weapon in dramatically changing the production of vaccines and other drugs. His passion for his research is inspired by his life experience. He says, I was born in India, and I have seen children die of cholera. Again, we're working on a cholera vaccine. Danielle is attempting to make vaccines and medications cheaper and more accessible for countries like his native India. Many of these treatments are not available, and they haven't been for decades, to people, but globally 1/3 of the population earns about $2 a day. Daniel uses a gene gun to fire human genes into plant cells, and when I listened to this, it was, he was firing, human insulin cells into these lettuce plants, okay? And then from the lettuce plants, they were expressing human insulin that then they were grinding up and putting into powder form into capsules and then feeding to people. And again, like I said, I mean, there isn't anything wrong with trying to make some sort of treatment. I mean, insulin, I believe in the past they have made through animals, for things such as thyroid hormone, they make through pigs, where they actually grind the pig thyroid to get the hormone or whatever it is, and then they make it in a pill format. But on the other hand, I mean, they're genetically modifying these lettuce plants and adding human genes to then get plants to express human genes. I mean, this is definitely mad science. This is playing the Isle of Dr. Moreau, and then they're going to give it to people to see what happens. Danielle's lab also produces vaccines against infectious diseases, for example, polio, tuberculosis, and malaria, and the production method is very different from what is currently available. Take polio, he says. And this was a quote that was pretty interesting. There's been a polio outbreak in Southeast Asia, and it has jumped to Africa. And unfortunately, it was vaccine-derived, he said. He added that the outbreak was blamed on vaccine storage. So again, this is this refrigeration issue. Because correct refrigeration was not provided, and the virus in the vaccine mutated. Now, the fact that he said that, and we had just read not one but two stories about polio mutating within people and then being spread through fecal matter. Do you remember that? That was last week. Into the sewage system, and they think that people with, and this is what they were saying in those stories, people with suppressed immune systems that aren't able to produce full antibodies allow the polio virus to live within the body and then mutate. So this story is very much contradicting these outbreaks, but it's very interesting that they're both saying that the virus has mutated because of the vaccines, regardless of what the issue was, whether it was the person's immune system or the vaccine wasn't quote-unquote correctly refrigerated. It has allowed the polio virus to mutate within people's bodies and spread it because they got vaccinated. So ding-da-da-ding, ding-da-ding-ding. It has pretty much been established now, and this is by another high-level scientist, that the polio that is now back in the wild was actually caused from the polio vaccine, and the polio is mutated within the bodies of the vaccinated, not all of them, but some, enough for it to now become an outbreak again, so now they have to be developing new vaccines. Danielle says that the vaccine-containing capsules won't need refrigeration, which makes them much easier to use in countries where keeping drugs cold is a problem. Once the lettuce plants grow and go to seed, the drug or vaccine-carrying lettuce could be regrown easily. So that's even scarier. So now let's just keep growing this genetically modified vaccine-producing lettuce. No, I don't know. Put it in your salad daily. It'll keep you healthy. That's a frightening thing. The production capacity of this, for example, for our anthrax vaccine. So they're working on one of those as well. And that was part of the DOD. We talked about this at Texas A&M University. They were doing anthrax vaccines with tobacco. Apparently they're doing it with lettuce as well. 1 acre of a plant can produce 320 million doses of an anthrax vaccine. So 1 greenhouse per disease, he says. That's really a frightening thought. Danielle said that he had just returned from a visit to what? Oh, a pharma company. And they showed me that these huge columns that they had to use to purify proteins that cost $10 million each and can only be used once, their whole facility costs $900 million. And all we need is a greenhouse and a place to make capsules. These guys are about ready to freaking bank. And then he kind of leaves the end. Let's reduce it down for the people who just don't get it. Danielle likens this potential cost reduction to another field where practices have dropped dramatically, and that is communications technology, and that is where I came from, me, myself, and I. Quote, and I remember these days, The very first phone call that I made when I came to the United States from India, it cost me $70. Now I make them free via Skype. That is a technological revolution, he said, and medicine needs that. Da, 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 da. So let's liken the simplicity of injecting, like, let's say, human genes or anthrax or polio into plants, and then having people eat them, to how easy it is now to call India via Skype. Okay. All right, now we're going to get to Dr. Mercola. And I got to do this fast because I wasted all that time in trying to get that clip. Hopefully this one works. Dr. Mercola, we're going to do the first just 3 minutes of this. He interviews a former nurse, 30 years in the field, talking about GMO vaccines utilizing DNA.
Welcome, everyone. This is Dr. Mercola, and today we are joined by Vicki DeBold, who is the NVIC's Volunteer Director of Research and Patient Safety. Professionally, she's trained as a critical care and home health care nurse and has more than 30 years' experience. She also holds a doctorate in nursing and public health. She also just finished serving 4 years as a consumer member of the FDA Vaccines and Related Biological Products Advisory Committee, where GMO vaccines were a topic of conversation during several meetings. All right, so you've served on this FDA committee for the GMO vaccines. I'm wondering if you could expand on just exactly what a GMO vaccine is, give us a little background on how you first became interested in this, and enlighten us about this emerging topic.
Well, there are a number of different genetically modified or genetically engineered vaccines out there. Some of them are quote-unquote naked DNA, where just a piece of the DNA of the organism that causes disease is introduced into the body. And then they range from different types of DNA vaccines to what's known as recombinant DNA vaccines, where the DNA of the pathogen is spliced onto the DNA of another organism, which is then grown in yet cell culture, so that the DNA issues that we're dealing with can be quite complex. So there's a lot of variation and variety in the new technologies that are being used.
And is there any danger inherent to this whole process of creating these new DNA vaccines?
Well, I think there's a lot of danger because we don't know what portion of the DNA can be incorporated into our own genome. We don't know what portion could be heritable to our children. We also don't know what happens when the immune system is exposed to DNA that has been recombined in lots of ways that the human body, through the course of time, has never had any exposure to. What diseases of the immune system may occur because of these exposures.
Is there, from your perspective, is there any risk or danger of these genetically engineered foods or vaccines to human health from what you've seen and reviewed in the literature?
Dr. Mercola, I think that there are risks to human health that have not yet been understood, and they come from issues related to the DNA of the pathogenic virus. It comes from the DNA that the virus may be spliced onto. In the case of flu vaccine, it's spliced onto something called baculovirus, and in the case of hepatitis B vaccine, it's spliced onto yeast. It comes from the cell cultures that are used to grow the vaccines. The new flu vaccines will be grown in armyworm cells. And it also comes from the fact that we have to use an adjuvant in genetically engineered vaccines to make them work. So there are lots of opportunities for DNA-related threats to human health, as well as threats from the chemicals that are used in the adjuvants.
This is Vicki DeBold. Says if you've ever had qualms about eating genetically modified foods, you will likely be deeply concerned about receiving a genetically modified vaccine as well. Such vaccines have already been produced, and some are even on the U.S. Center for Disease Control and Prevention's recommended vaccine schedule. And we talked about some of those, one being the Gardasil vaccine with those virus-like particles. Even though, and as is the case with GM foods, we know very little about their long-term effect. Vicki Diebold, PhD, RN, Director of Research and Patient Safety with the National Vaccine Information Center, spoke with me about the many reasons to be wary of this new technology, which is far more intertwined with biotech innovations like GM food than you might think. Says nobody knows what happens when you inject people with GM vaccines, and as we've established, nobody knows what happens when you give people edible vaccines either that are transgenically made. Says there have been some fair warnings, though. In 2006, researchers wrote in the Journal of Toxicology and Environmental Health, quote, genetically modified viruses and genetically engineered virus vector vaccines possess significant unpredictability and a number of inherent harmful potential hazards. Horizontal transfer of genes is well established, and new hybrid virus progenies resulting from genetic recombination between genetically engineered vaccine viruses and their naturally occurring relatives may possess totally, and this is the key phrase, totally unpredictable characteristics with regard to host preferences and disease-causing potentials. There is an inadequate knowledge to define either the probability of unintended events or consequences of genetic modifications, and that's absolutely true. I mean, go down a generation or two and we have no idea what the stuff is going to do. Back into Dr. Mercola's article, though this was 6 years ago, little has changed, and even as the technology has advanced, today we have several different types of genetically modified vaccines in production, development, or research phases, and this story was from 2012, so just know, add another one to this, and this is edible genetically modified vaccines. But back in 2012, we had the DNA vaccine, and that's DNAs for a microbe's antigens are introduced into the body with the expectation that your cells will take up that DNA, which is then instructing your cells to make an antigen molecule. And as the National Institute of Allergy and Infectious Disease, which is a division of the National Institute of Health, put it, quote, in other words, the body's own cells become vaccine-making laboratories. And I have a full story about that because DARPA has jumped into that game. The second type of vaccine, GMO vaccine that they had in 2012, is called a naked DNA vaccine, and a type of DNA vaccine in which microscopic particles coated with DNA are administered directly into your cells. And the third one is recombinant vector vaccines, and this is similar to DNA vaccines, but they use a virus or bacteria to act as the vector or carrier to introduce microbial DNA into your cells. So I've seen them doing that with yeast, now in some of this new stuff as well as virus or bacteria where they suck out the nucleus and they put other things in there. It says there are experimental GM vaccines being developed that use tumorigenic cancer cells and cells from humans, dogs, monkeys, cows, pigs, rodents, birds, and insects. And you know, we had read the other day as well about some of this cow protein, and I've seen it on the labels. It's called bovine serum. That now there's a concern that that bovine serum, if that came from an infected cow with BSE, more affectionately known as mad cow disease, that could potentially spread some malformed proteins within the receiver of that vaccination. And that was in one of the, you know, who knows, peer-reviewed journals, scientific journals that we read from a day or two ago. Anyway, back to the story. What happens when foreign DNA is inserted into the human body is a mystery. There was a story from last week that we read from a woman who was doing a study based upon human fetal stem cells being used, and she had found evidence that the DNA fragments from that fetal stem cell tissue that is also being used, and I believe it's the MMR vaccine, can be absorbed into the human DNA, because it is human DNA. So human DNA may absorb human DNA. She has evidence for that, and cause mutations within, again, the receiver of that vaccine. So guys, this is honestly leading edge mad science, and we do not have any type of long-term studies on the stuff that is already being trialed in humans. Back to the story. When will it trigger undesirable changes in human cells or tissues, and will it combine and exchange genetic material with human DNA? Well, again, this story was from 2012. We have research from 2015 that shows, yes, that it very well may. Will it transfer to future generations? Well, if any of the stuff that we've read about epigenetics and our, our microbiome and how all this is interconnected, if that's any indication, even how, like, our bad experiences and being depressed and, bad lifestyle choices can be passed on to our progeny, I'm gonna have to say emphatically, very much probably, yes. So when we see videos that came out from RT back last year about, in 30 years, we're going to be a new human species, human 2.0, remember that one? I must have played that video about 20 times. You can see where that is coming from. And then you see lunatics like Ray Kurzweil mention in an interview that the whole process of being assimilated into this new generation of transhumanists has already started. You know that it's game on here, Charlie. This isn't good at all. All right. I'm not going to get to everything I wanted to get to today, which is very sad, but hey, we'll keep going. We'll make this a whole week of crazy transgenics. It says what we don't know is that a portion of the DNA might be incorporated into our own genome, and we don't know what portion could be inherited to our children. We also don't know what happens when the immune system is exposed to DNA that has been recombined in lots of ways that the human body through the course of time has never had any exposure to. What diseases of the immune system may occur because of these exposures, said Diebold. And that's exactly it. I mean, we have this major outbreak of autoimmune diseases where our immune systems, and I'm one of those people, they told me about 20 years ago that I was immunocompromised. And I've had to deal with this whole progression of weird symptoms that I've been able to trace back to the early 90s. Two things that happened. One, I received a flu vaccine back in college because everybody should get one, especially when you lived in the dorms. And the second thing was the weird reactions to the tomatoes that I had in the early 90s. And then we find out that they had these ready-served GM tomatoes out there in the food chain, and then we find out that, they'd been experimenting with vaccines in tomatoes, so who knows? I mean, honestly, they pick little pockets, and they do experiments, and we established that looking back through the history of the biowarfare program of the United States, and all these horrifying experiments that have been done on the populace without their knowledge. Anyway, back to the story. Quote, and this again is the guest, Diebold. She is a PhD, RN, 30 years experience. Use of foreign DNA in various forms has the potential to cause a great deal of trouble, not only because that there is potential for it to recombine with our own DNA, but there's also the potential for it to turn the DNA switches. And how many times have we talked about this? This is this lunatic RNAi technology. There's the potential for it to turn the DNA switches, the epigenetic parts of the DNA, ding, ding, on and off. So we've been right on the money the whole time. This is exactly what they're experimenting with. And let's add to this lunatic mix Monsanto and all their new technology for quote-unquote DNA insecticides, flipping protein switches on and off inside the bugs who ingest it to slowly kill them as they starve from not being able to produce proteins that are necessary for biological processes. All right, let's continue on because I am quickly running out of time. There's an article that came out of HHS Public Access, which is Health and Human Services, which is a division of the National Institute of Health. titled Oral Delivery of Human Biopharmaceuticals, Autoantigens, and Vaccine Antigens Bioencapsulated in Plant Cells. Yes, it's just everything we started the show out with, and this was published in the final edition as of, the original one was October 23rd, 2012, and then it was modified June 15th, 2013. Abstract, and I think we'll just read the abstract because it is very long, but I'll link to this for the full document. It's a 4.3 megabyte PDF in the description of the show. Abstract. So this is something that, if it's National Institute of Health, I'm sure this is the U.S. National Library of Medicine of the National Institutes of Health. You know that there is some federal dollars behind this. Among 12 billion with a B injections are administered annually, unsafe delivery leads to greater than 20 million infections, which is a staggering number, and greater than 100 million vaccine reactions. What? And this is in a National Institute of Health publication? They're like fully admitting that there are greater than 20 million infections that came from quote-unquote unsafe delivery of the old school vaccines, because it was because what? They weren't refrigerated? Because I think that that's going to be the new mantra to get around what has been a failure. And 100 million reactions. So that explains the VERS database, doesn't it? It says in an emerging new concept, freeze-dried plant cells, in this case lettuce, expressing vaccine antigen / biopharmaceuticals, are protected in the stomach from acids and enzymes, but are released to the immune system and the blood circulatory system, where plant cell walls are then digested by microbes that colonize the gut. They are literally using your microbiome to release vaccines. Your microbiome, we found, also is connected to your vagus nerve, which is a direct connection to your brain. This is giving them direct access to your brain. Vaccine antigens bioencapsulated in plant cells, and that's your brain via chemical interactions, let's say. So they can turn chemicals on and off in your brain using chemical reactions. Vaccine antigens bioencapsulated in a plant cells upon oral delivery after priming, conferred both with mucosal and systemic immunity and protected against bacterial, viral, or protozoan pathogens or toxin challenges. Oral delivery of autoantigens was effective against complications of type 1 diabetes and hemophilia by developing tolerance. And oral delivery of pro-insulin or extendin 4, so this is that insulin one that was in that interview we didn't get to hear, expressed in plant cells, regulated blood glucose levels similar to injections. Therefore, this new platform offers a low-cost alternative to deliver different therapeutic proteins to combat infectious or inherited diseases. What? To combat inherited diseases by eliminating inactivated pathogens. So, I mean, are they going to use this to flip switches in our DNA? And they're going to not only introduce pathogens, but they're going to do other things. I'm telling you, all this technology is so beyond mad science, they could turn us into whatever they want when they have access to our full DNA sequence to be able to manipulate and change based upon whatever they wanted to breed into this food. Combat infectious or inherited diseases by eliminating inactivated pathogens, expensive purification, cold storage and transportation, as well as sterile injections. And this goes on and on. I'm going to tell you, this is a very long document that kind of goes through all the different ways of production, the mucosal delivery, how easy it's going to be to put this in a capsule or mix it into the food. And we will all be vaccinated, and the world is going to be a much happier place. They can turn us into whatever type of creature, mind-controlled zombie they want. And if not to add insult to injury, September 11th, how fitting, 2015, out of livescience.com, DARPA, cue up scary music from Star Wars, DARPA is developing human biofactories to brew life-saving vaccines. And guys, this is using some of the same technologies. The next factory for life-saving drugs could be the human body itself. Scientists are developing a new vaccine-making method that co-opts, yes, co-opts the human body's ability to quickly create antibodies, its main weaponry for fighting disease, says researchers with DARPA. Now, when you think about the ability to co-opt the human body, in this case to make antibodies, but I don't know, in another case to turn off proteins, in another case to turn off thinking abilities, to stop producing serotonin or melatonin, to stop producing insulin, to stop producing, you know what I mean? Like, all these things, once they've cracked that genome to be able to flip switches on and off, we were completely at risk. You could be killed in an instant. And it could be done through your food supply. I mean, it's become a reality. And now that they've gotten rid of all the labeling, and they're doing it on a global basis, and let's not forget about, what if some terrorist group in another nation, quote unquote terrorist, some rogue nation, gets a hold of this technology, and since nobody's inspecting the food that comes in anymore, something like this gets inserted into your who the hell knows what. Freeze-dried lettuce into your super greens that were imported from, Pakistan. I don't know. Just saying, this is to me, when I start reading about this stuff, it literally makes me dizzy. And it makes me dizzy because it makes my blood pressure go up. And it makes my blood pressure go up because my brain just starts firing off all these thoughts as to how this could be used. This is a horrifying, horrifying development in the field of biotechnology. It says this new method of vaccine production would involve giving the body instructions for making certain antibodies. And because the body would be its own bioreactor, the vaccine could be produced much faster than traditional methods, and the results would be a higher level of protection, says Colonel DOD Daniel Wattendorf, a clinical geneticist with DARPA. Just what we need, more clinical geneticists that work for mad science gone mad, top of the food chain, which is the branch of the U.S. Department of Defense charged with developing new technologies for who? Oh, yes, the military. Wattendorf described his research here at the Wait What? Technology Forum hosted by DARPA. It was a conference that just ended somewhere. I don't remember where it was. The current process for producing vaccines could take at least 9 months. And when what? Yes, let's pull this one out of the archives. The H1N1 swine flu pandemic spread in 2009. Researchers grew the vaccines in mouse ovaries for months, which I remember reading about that. Like, growing vaccines in mouse ovaries, even worse than chicken eggs, before they could produce sufficient quantities of the drugs to, yes, administer it to people. Thank God it was a pandemic, because we had to get that stuff out there under Project BioShield. But ideally, the body will mistake the vaccine, essentially a harmless protein or antigen, from the outer coat of the virus for the virus itself, and in this case, of mistaken identity, then spurs the immune system to produce millions of antibodies, which latch onto and neutralize the invader. Unfortunately, in the 2009 outbreak, only 40 to 60% of those who were immunized developed antibodies. Now again, this seems to be the first time I've heard them publicly admit that, our last big vaccination, so we heard about the polio vaccine failing en masse in a publication by a reputable doctor. And now we're finding out that The swine flu vaccine of 2009 also failed en masse because they got everybody to jump on board and get it. And oh my God, it's a pandemic. But it didn't work because what? 40 to 60% of the time it failed. Oh no! Problem, reaction, solution. So all this science that we've been pointing to the tinfoil hat wearers, going, you guys are anti-science, you don't know what you're talking about. We now have scientists, DARPA, coming out and saying, our vaccines don't work. But hey, don't worry about it, because guess what? We already have a solution. That's DNA vaccine, recombatant mad science grown with plants, injected with animals, a sucked out nucleus, and then putting it into your food so it manipulates your DNA and it'll cure you. Yes, that's our solution. When scientists crunched the numbers, they found the glacially slow production process plus the limited vaccine effectiveness meant that the 2009 influenza vaccine protected just how many? 1.6% of the 60 million plus Americans who got the shot. But don't worry about it, because you know what? We were growing it in mouse ovaries, and none of you guys grew an affiliation for cheese, even though we gave it to 60 million people, and it didn't work. Oh my God, that was a massive human real-time experiment. And then, what happened? Ebola, bleeding out your eyeballs. Yes, so the Ebola vaccine, a new Mad Science Gone Mad project that it's an emergency we get to again test in people. But last year, the Ebola crisis points the way to a potentially faster method after a medical missionary, Dr. Kent Brantley, successfully fought off the deadly virus following a plasma transfusion from a patient. He returned the favor by donating his blood to other infected Americans. But because his body had vanquished the virus, it was teeming with Ebola antibodies. And what they did was, because of this good Samaritan, they were able to take his antibodies and create a quote-unquote vaccine. So scientists would harvest his viral antibodies, and then they would isolate the most effective ones, determining the genes needed to make that antibody, and encode many copies of those genes into a circular snippet of genetic material, either DNA or RNA, that the person's body would then use as a cookbook to assemble the antibody. In this case, they used a single needle jab, and they would insert the genetic antibody recipe into someone's muscle cell, and once inside that muscle cell, free-floating RNA would latch onto DNA or RNA instructions and make many, many more copies of the antibodies, and using RNA to encode instructions for making the antibody would quickly ramp up antibody production to an effective dose within your body within a few hours. But the protection may dissipate quickly. DNA takes a day or two to produce antibodies and must be inserted via a more painful process called electroporation, in which the antibodies will continue to circulate for months. Wattendorf says the body itself has become a bioreactor. So far, a number of different companies and institutions are developing potential vaccines using exactly this method. But unlike gene therapy, the genetic instructions for making the antibody are not encoded into a person's genome permanently. Thank goodness, he added. Instead, the genetic instructions for making the antibody gradually degrade within your body over time. And guys, we're going to have to leave the horror story right there and pick this up tomorrow, because we're down to about the last 15 seconds of the show. Today is Wednesday, so make sure you stick around. Ms. Lonnie Clark is up next. Thank you again, Lonnie, for helping out UCY. Then we have Nuclear Hot Seat with Libby Haleve. Make sure you stick around. Josh is live at 6. The boys from New York live at 8 p.m. Eastern. Truth or Talk with April and Burstein. We have the whole interview at 9, and Anthony with Nick at 11. Have an awesome Wednesday. We'll catch up with you tomorrow. Take care.
think the use of foreign DNA in various forms has the potential to cause a great deal of trouble. Not only because that there is the potential for it to recombine with our own DNA, there is the potential for it to turn the DNA switches, the epigenetic parts of the DNA, on and off. So I think that we're still in a place where there's a lot of work that needs to be done to figure out what the short and long-term effects of this type of DNA are on people who have lots of different genetic makeups.
Maybe if we could just expand on your concern for a bit with this recombination of the genetic material and help us understand how that would work. So you've got this edible vaccine, for instance, that is some type of plant material typically that's grown with the new introduced engineered vaccine component. So, and that typically is a strand of DNA or some nucleotide sequences that represent the antigen they're trying to build a response to. So how is this combination going to occur? I mean, what's your concern? And I just want to have everyone understand it a little better.
Okay, well, this is one of the delivery technologies that are being explored, the new using vaccines orally, meaning what you eat and ingest, and that would involve putting hybridized or somehow modified DNA for pathogenic virus or bacteria into food that's eaten, and then it would be ingested and then incorporated somehow to the bloodstream so that the immune system reacts to it. There are other ways of introducing genetically modified vaccines that we need to be aware of as well. Besides the standard injections, there are nasal routes, squirting vaccines up the nose, and then there are the transdermal routes that I think are very important because they involve use of nanotechnology, which would permit introduction of DNA directly into our cells. Genetically engineered vaccines are already being used. Beginning in 1991, the hepatitis B vaccine was licensed and is given to infants beginning on the first day of life. It is considered a genetically engineered vaccine. It's a recombinant vaccine where parts of the hepatitis B virus are recombined with yeast. This is one of several vaccines that are already being given to infants and teenagers and are part of the CDC's recommended vaccination schedule.
Are there any recommendations you have for the person watching this with respect to how they might make an impact on this type of movement progressing much further, the introduction of genetically engineered vaccines?
Well, I think it's incumbent upon everyone, as usual, to be educated, to read and find out which vaccines are genetically engineered and what they're not, and to demand of the scientific and government communities that they do the type of safety testing that we need in order to make informed decisions. Be involved, understand what's going on, understand what can be done in terms of asking for better evidence of safety and effectiveness. One of the best ways to be involved is to go to nvic.org and sign up for the advocacy portal, where we are actively working on a number of vaccine issues, and genetically engineered vaccines are part of the topics that we're trying to stay on top of as states begin to make changes to their laws. All over the country, the right to choose and to exercise legal exemptions to vaccination is being challenged in state legislatures. And it's really important that people be educated and aware of how the laws are changing, what that means for being able to give informed consent. And the genetically engineered piece of it is critical. It's very critical that people understand how the laws related to not just genetically engineered vaccines, but genetically modified foods of all sorts have the potential to affect human health in ways that we absolutely do not understand at this point.